Hi everyone, my name is Michael. I am a second year biophysics student. I joined the June lab at UC San Diego very recently, and we've only been working with these cells for a month or so, but I'm excited to show some early data that we have. So for those who don't know us, oh, great, okay, there we go. For those who don't know us, we do biophysics. In general, we look at bacterial physiology. So we look at the cell cycle and things like E. coli, B. ceteris. Now we're looking a little bit into yeast as well, and I guess mycoplasma now as well. Generally, one of our primary kind of experimental techniques is the mother machine, which is a microfluidic device. You have probably seen these, but I've got some kind of footage from one where basically we load E. coli into some kind of channel, and then we have some kind of strong fluid flow up top, providing some kind of nutrients. And we can watch cells grow for very, very long periods of time under very tightly controlled conditions. Some of our theory work relates to things like entropic springs and chromosomes, and we've used these mother machines, these almost like pistons before. So that's the kind of stuff that we do. Okay. So our goals with this mycoplasma, generally, it would be good as kind of a preliminary step to just grow these in the mother machine under different kind of widths and so on. From there, we're kind of looking to use them as a tool to understand how cells control what size they are when they divide, and potentially other more complicated organisms like E. coli. So experimental results, this is just like an agripad, an early agripad from like Syn 3.0. Again, we started a month ago, and also I am a physics student, so some senior levels of the lab had some comments about my data quality. But yeah, I'm excited to show you guys some early data. So this is Syn 3A growing in a mother machine. Here we have the fluid flow up top. We have channels that are roughly 0.5 micrometer in width. We've loaded them with Syn 3A, as you can see, and they're growing up, and then they start pouring out of the mother machine. So this was just kind of a preliminary show. The next thing we did was we grew JCVI Syn 3.0 in a very large mother machine. This one is something like 5 micrometers in width. And we got to see some of the kind of characteristic behaviors of Syn 3.0 pretty directly. So here we see these filaments that are, I think we observed them in a lot of different experiments, but it's really nice to see them kind of dynamically and growing like this. Here, for example, because there's this flow coming up top, you can see that it induces some kind of shearing force, and it kind of spins the the filament into this little spiral. All right, and then we also have some footage of sort of bubble formation in this Syn 3.0. We're not really sure what the bubbles are yet. I'm actually extremely interested to hear what people have to say. Where essentially, as they grow, you can see these spontaneous bubbles arise among all of the mycoplasmas. So you can see it right there. Yeah. All right. So yeah, really short talk. Once again, I haven't been working on these for very long. Thanks to John for helping us get started and making some media for us. Sakchun for letting physicists try his hand at biology, and the lab for being wonderful and encouraging. All right, great. That's all I have.
Michael, I'm dazzled at the progress you guys have made. James Pelletier, I think you ought to be the first commenter here.
Hi, Michael. This is James Pelletier from the National Biotechnology center in Madrid, and I was an undergraduate in the lab of Sakjun a number of years ago, and a huge admirer of the work that your lab is doing. And congratulations on your fantastic progress in such a short time frame. It's really exciting to see the cells growing in the high confinement, the 500 nanometer wide channels that we'd never explored cells growing in this kind of confinement. Um, so that's really exciting, and I'm looking forward to learning more from your work. I think one question that I have would be regarding the imaging conditions. So so far you've looked at the cells in bright field. Have you tried any kind of fluorescence microscopy?
No, I haven't had a chance to try fluorescence microscopy.
But
I,
you have the string with M Cherry, don't you, Michael?
yeah, we do have I think Syn3B with M Cherry, but I haven't had a chance to grow it yet.
Okay, I think it's Syn3A that you have, but that's okay.
Okay.
And you should also have Syn1 if you need, but let me know.
Okay.
This is, I can't wait to see how this develops. You've got a tool here that I think more people can start thinking about as they get exposed to what you can do with it.
Yeah, I think the experiment I'm probably most excited to run is putting the Syn3O in some kind of like seriously confined channel, like maybe 500 nanometers or a little wider, but I haven't had a chance to do that one yet.
And then the bubbles that you talk about, I think, and I think James will agree with this, that these are vesicles that the cells make. So, mostly they do not have cytoplasm in them, but sometimes they do, and sometimes they have DNA in them. But
I agree with
I hear
that, John.
someone speaking. James, is that you?
Oh, that wasn't me. But separately, I agree with what you were saying.
And a lot of bacteria make outer membrane vesicles as maybe a defense mechanism, but it could be that something in these cells in Syn1, we have we are making more membrane than the cell needs. But we don't understand that yet, and this is something that we're excited to try to figure out at some point.
Okay.
Other questions for Michael? It's great to get more physicists involved in this.